Thanks everyone for coming. We're delighted today to have Professor Mark Lipsitch and Professor Derek Smith speaking. Pandemics have been one of the biggest things to worry about for the human species in the 20th century. In the 1918 flu virus killed more people than the First World War did, so they're better at killing us than we are ourselves. In the last couple of years, there's been a very interesting debate happening over a particular set of experiments on transmissibility of influenza. One of the things that makes flu particularly worrying is that there are new strains always emerging, and any information that we can get about what strains are likely to develop the characteristics to be transmissible in humans is very useful in our response. However, some prominent voices within the scientific community have said that certain experiments that look at developing characteristics like these in a laboratory setting in order to study them may carry inherent risks. For example, if there was an accidental release of an organism that people were working on. And Professor Mark Lipsitch has been one of the most prominent voices raising these concerns. He was one of the founding members of the Cambridge Working Group, which is a large body of scientists who have raised these concerns. We're very lucky as well to hear from Professor Derek Smith, who has spoken eloquently about the reasons in favour of doing this kind of research. From a CSER perspective, this is particularly interesting for a couple of reasons. One is that the scientific debate, the scientific community seems to be split at the moment, with very credible experts raising concerns on both sides. And the other is that there are real reasons for concern about developing a line of scientific experiment that may carry inherent risks. However, we should also consider that if we shut down inappropriately a line of experimentation that has the potential to save many, many human lives, we're taking on another risk that's equally as valid. So without further ado, I'd like to invite Professor Mark Lipsitch to begin, Professor of Epidemiology in Harvard. Thank you.
Thank you, Sean and Hugh, for the kind introduction, and thanks to everyone for coming. I think this is, it was a helpful introduction in a number of ways, one of which is the more general concern about synthetic biology, which I think a number of people share as both for its potential benefits and its potential risks. I think this might be a paradigm case of an early form of synthetic biology, which in some ways is much easier, and I'll try to explain what I mean by that, both to evaluate the risks and to evaluate the benefits. And I think the stakes of getting this discussion right, whatever that means, are relevant for the discussion itself and also for our future thinking about synthetic biology and other types of work. So I'm going to talk about a fairly limited set of experiments and their risks and benefits, those experiments being the risks and benefits of gain-of-function experiments on potential pandemic pathogens. A few disclaimers. These are my own views. I hope some of them will come to be your views, and they are to some extent shared by many of my collaborators with whom I've written articles expressing some of these views, but this is my own take. And it is also not necessarily the views of the NIH, which is my major funder, and which is also, until the recent events that I'll tell you about, was a major funder of gain-of-function research. So I'll tell you what gain-of-function is in a minute, but the term that I prefer to use actually in describing this class of experiments that are of particular concern is potential pandemic pathogens. That term, to my knowledge, was invented by Lynn Klotz and Ed Sylvester. And as I use it, it means an organism which combines a pathogenic organism which combines three properties: transmissibility in humans, novelty in humans, and virulence in humans. And the reason why that particular combination is of concern is that transmissibility turns the virus, in this case, into something that we all have to worry about rather than something that the people in a particular lab have to worry about. Virulence tells you the scale of the worry that we make. And novelty means that we are, if a pathogen is novel to the human population, the likelihood is that there's very little or no immunity to it that would protect individuals against infection. So the class of experiments that I'm going to talk about is what's known as gain of function by many. And the idea of gain of function is a common idea in microbiology and in other sciences, biological sciences, that it's experiments in which a particular organism or cell line gains the ability to perform some function. But and nobody has any question that that's an important technique that we all need to use in some biological experiments. So the concern is specifically on gain of function when the function being gained is pathogenicity in a transmissible organism or transmissibility in a pathogenic organism, in this case a virus. So the paradigm or the kind of classic setup for one of these experiments, as has been published in the last few years, is to start with an influenza virus, to do some combination of direct genetic manipulation of that virus and selection in experimental animals. In this case, ferrets are the most common experimental animal, by which the virus is passaged from one animal to the next with the goal of facilitating the evolution of its ability to pass on its own without human intervention, simply by aerosol transmission from one ferret to the next. Ferrets are thought to be probably the best or one of the best models for human infection and disease with influenza. Those viruses, as they're passaged, are then sequenced and their phenotypes assayed, for example, their ability to bind to particular receptors on mammalian cells. And then that process is repeated so that the course of evolution and adaptation to the mammalian host can be monitored. And the two really studies that got this whole discussion started were both funded by the United States government, which will become relevant in a minute, but were done, one in the Netherlands and one in Wisconsin, and were done with H5N1 influenza, the bird flu that we all heard about from the late 90s to the beginning or the mid 2000s. It's still circulating in birds in many parts of the world and occasionally infecting humans, but we hear less about it, partly because other flus have emerged to take the spotlight. But those are not the only ones. There have been quite a number of other experiments published in various places by various groups and funded by various funders, both public and private. I will say that the Gates Foundation has explained that they are no longer in the business. They funded the first of these experiments, but have stated that they don't intend to continue. But the other ones have been funding this kind of work at various levels for some time. More on the funding in a moment. But just looking at it from a broad perspective, the kinds of experiments that I'm describing are similar in their potential for mishap to, I would argue, the most dangerous kinds of experiments that we have going on in any laboratories, biological laboratories, in the sense that they are creating strains that have high virulence, that have transmissibility and pandemic potential, and are novel. So they fulfill those three requirements of a pandemic pathogen. And the only other type of work that I would say matches that is work on smallpox. Smallpox work is controversial whether it should continue or whether indeed the existence of smallpox should be perpetuated or whether it should be destroyed. But at the moment, the work is done in 2 laboratories, one in Russia and one in Atlanta, and it is done with very strict controls over what kinds of experiments can be done. There's an international committee run by WHO that has to vet these experiments. If the quantity of virus being manufactured is too large, for example, that's a reason not to consider doing the experiment. So there's very stringent control. And by contrast, the work that I am describing is done in not the highest biosafety level and not with those kinds of controls, but in sort of an intermediate between the highest and the next to highest, so-called biosafety level 3 enhanced or 3 plus. By contrast, routine work with characterizing either highly pathogenic flu viruses with highly pathogenic flu viruses that are not transmissible is done in biosafety level 3, so a half step below, and work with, for example, filoviruses is done at biosafety level 4, so a half step above. And I would argue those are much less dangerous experiments because of the relative lack of transmissibility of those viruses. So there's a mismatch between the level of security and safety we require of these experiments and their risk. All of this sort of came to a head over the summer of 2014 when a series of things happened in highly visible US government labs. The first was that smallpox, thought to be destroyed in all places except Atlanta and Vector in Russia, was discovered in a storeroom along with a number of other pathogens in vials. Nobody was exposed to them, but it was newly discovered at a freezer in the NIH, FDA, NIH. It was a shared facility. Soon thereafter, there was a removal of what was thought to be killed anthrax bacteria from a biosafety level 3 laboratory in CDC to a lower level containment laboratory where they were treated in a way that could create aerosols. The decontamination had not been complete, and something like 7 dozen or so workers were treated with antibiotics for possible exposure. And while CDC was investigating this, it was discovered that another mishap had occurred also I think in the spring of 2014, in which contaminated stocks, contaminated with a controlled agent, a select agent, H5N1, had been sent from a CDC lab to another lab that was not expecting to receive it, although it was actually equipped to receive it. More recently, to break chronology briefly, on Christmas Eve, just a few days before Christmas Eve of last year, it was discovered that CDC had switched a killed vial of Ebola virus and a not killed vial and moved the not killed vial into a low-level containment laboratory. We're continuing this pattern, which I think will become important when we talk about risks, of taking not just of exposing workers in a given laboratory, but of exposing workers in a laboratory that is not expecting to be exposed. And that's one of the potential scenarios that I think is of particular concern because it's so much harder to control and to mitigate if it happens, because it's unexpected. I wrote a piece that appeared in the New York Times over the summer suggesting that the anthrax experiments were a warning of the sort of thing that we should be concerned about if a transmissible pathogen was being created in a laboratory. And then in the middle of the summer, a group of us were meeting in Cambridge, Massachusetts, to discuss how we could address this problem in general. It was concerned people, scientists of various stripes, and it was the weekend. It was the Monday after the weekend when many of these events had been announced, and some other events related to them had been announced. Everyone's cell phones started ringing in the room, practically, from reporters who had done what they were supposed to do with a Friday afternoon announcement and ignored it. So the government succeeded in doing what it meant to do by burying the story. But on Monday morning, they woke up and started pursuing it, and everyone was being asked what they thought, and we decided that as a group we should make a group statement, which we did, and which stated that which called for the curtailment of this kind of work until a comprehensive risk and benefit assessment could be done. Soon thereafter, a group calling itself Scientists for Science formed. They were ostensibly in opposition to our Cambridge Working Group statement, although there were several people who became members of both groups because there was actually a lot of overlap. I actually probably in literal content, if not in tone, would be willing to sign on to the Scientists for Science statement because there is a lot of overlap, which tells you something about the power of consensus to mitigate outlying views. But in any case, Scientists for Science emphasized the benefits of doing work on this, on all dangerous pathogens, which we agree, but in particular on these generating these transmissible pathogens, whereas we singled those out as a particular area of concern. Fast forwarding to October, there was an announcement from the White House that the U.S. government, which, as I said, had been the major funder of such work, was going to pause funding on all such work for a year while a risk and benefit assessment was done. That funding pause generated letters to recipients of 18 grants from the National Institutes of Health. It was government-wide, but all of the relevant grants were through the National Institutes of Health. It was 18 out of approximately 250 grants that the U.S. government was funding at the time on MERS, SARS, and flu. We didn't expect MERS and SARS to be in it. And in fact, those two coronaviruses, which are not related to flu but have some properties in common, were removed from the funding pause in December. So now about 12 grants are affected out of about 230 grants on flu. So what I want to talk about is something about the risks, a little bit about the ethics, a brief discussion of the benefits and of these experiments, and then some discussion of alternatives. So I'll give away the punchline of each section, but actually before I do, I should say each of these arguments is independent, and it has been pointed out that it's sort of strange that those who emphasize the risks are also highly skeptical of the benefits, like me, and that those who emphasize the benefits also minimize the risk, like we'll see if Derek falls into that category. But those two cells of the 2x2 table are occupied, and there aren't very many people who say these are incredibly risky experiments, but they're worth it. And there aren't, well, they aren't worth doing and they aren't risky category is perhaps understandably fairly empty. But it is quite coherent to believe independent things and even contrasting things about the risks and the benefits. As it turns out, there are not many people in the world who have made their views known that seem to hold those. But I think you should be skeptical of what I say on and what Derek says on both fronts, but not necessarily come down on the same side of each of the issues. So in terms of risks, the claim that I want to make is that there are small but estimable probabilities of catastrophic outcomes, and that when you multiply the probabilities times the outcomes, the numbers are quite concerning, very concerning. And the numbers that I'm going to describe are a first stab. They are an effort to show that it's possible with reasonable support to make estimates of these probabilities and consequences. They're certainly not meant to be the final answer, which the U.S. government, at least, is trying to get from an independent party. They have been criticized and will be criticized further, and indeed I'll give away another punchline, which is that one of the proponents of these studies has taken the numbers that I will present to you in a minute and has proposed that the actual numbers are 7 orders of magnitude lower and not concerning. So there's a lot of white space between those two, and somewhere in the middle perhaps lies the answer, although I actually think the numbers that I present are on the optimistic side, but you can decide for yourself. But the point is really to establish that this is not incalculable, it's not impossible to figure these numbers out. There are enough data out there, and that even though the numbers are going to be imprecise, if you are told that the risk is between enormous and gigantic, you don't really care where it is, you just know that there's a very large risk. The fact of imprecision is not a problem. It's only a problem if the imprecision scales, but spans the boundary between concerning and not concerning, I would argue, in terms of making a decision. Of course, we'd like to reduce our uncertainty. So, as a general matter, laboratory safety has been a success story in the 20th century. And the way I mean that is that basically not many people have died from laboratory accidents. There's lots of underreporting, and there probably have been some deaths that we don't know about. But the number of known accidents is around 1,000 in a 25-year span, and the number of deaths is around 1 per year in that span. So it's very small. And this is not what I'm talking about. I would be kicked out of the Center for the Study of Existential Risk if I were talking about 24 deaths over 25 years. On the other hand, there have been a number of major breaches that have caused large, not existential, but large real risks or potential risks. In Beijing, I'm just listing the ones in Beijing there was a laboratory accident with SARS about a decade ago, in which there were 2 primary cases who went on to infect 7 others before it was controlled. There were similar events in Taiwan and Singapore, also with SARS virus, but there were no secondary cases in those examples. There was the famous foot and mouth disease release from Purebright that you're aware of if you've been in the UK since 2007, which caused major agricultural consequences. It's an animal disease, so it didn't cause human consequences. And probably the 1977 H1N1 strain that circulated for 22 years following that reemerged because of a laboratory escape, although that is not proven. The United States is well known for making these kinds of accidents, at least over the summer and up to Christmas Eve, but UK is also a world leader in this area. I don't want to be chauvinistic. Ian Sample has done a lot of work at The Guardian trying to uncover this. It turns out that, as in the US case, the known accidents seem to concentrate in one site. In this case, it seems to be Purebright, which keeps on having accidents over and over again, although none as consequential as the one that caused the 2007 outbreak. So if laboratory safety is not a big deal over the last 25 years, why am I talking to you? The answer is that in most cases, laboratory safety is a matter of occupational health. It's a matter of 1s and 2s and 3s of people who are exposed, potentially infected, and in a few cases get sick, and even fewer cases die. But because the organisms being worked with are not transmissible or are readily controlled, they present primarily or only individual risk. For example, anthrax cannot be transmitted from person to person. And even Ebola we thought even in almost anywhere, and now we know there are some exceptions, but even Ebola is not readily transmissible if it escapes a laboratory. On the other hand, when we're talking about a transmissible infection, we're talking about public health, not occupational health. And activities that threaten public health arguably should face much more stringent restrictions than activities that risk occupational health. So the point is not that work on dangerous pathogens should be curtailed. It should not be. It should be continued, and I would even advocate increased. The point is that the very small fraction of that work that involves creating novel transmissible virulent pathogens deserves extra scrutiny, and I would say should not be done. So, so how can we try to estimate this risk? It starts from the rate at which accidents like the ones in CDC happen, but also resulting in laboratory-acquired infections. So those accidents over the summer, fortunately, did not result in any laboratory-acquired infections. But in U.S. select agent labs, so these are legally controlled pathogens used in laboratories, there were at least 4 laboratory-acquired infections in, at most, 2,044 laboratory years. And I say at least and at most because of underreporting and some peculiarities in the way the data are shown, so that we have a lower bound of 0.2% per laboratory per year. And if you just look at the National Institutes of Allergy and Infectious Disease intramural labs, so the ones on the campus in Bethesda, Maryland, and you look in terms of full-time worker years, it's about 1% per full-time worker year. So for every 2,000 hours someone spends in a BSL-3, there's about a 1% risk that they get infected, historically. So if we're willing to define risk as the probability of a bad outcome times the consequence of that bad outcome if it occurs, then we can begin to make calculations. So I've just shown you the estimates for the probability of a pandemic, of a laboratory-acquired infection for a given unit of research, and we'll stick to the laboratory years estimate just to keep things manageable. You could make a similar calculation for the other ones. And the probability of a pandemic occurring, of course, is the really serious concern. And that one, we have to use models. We don't have data fortunately, to show the probability that any given laboratory-acquired infection leads to a pandemic. And that's because all of the laboratory-acquired infections that have been reported so far have been with relatively or completely non-transmissible agents. So they're handled at the same biosafety level, or nearly, not quite, the same biosafety level as the ones that I'm talking about, but they are non-transmissible. So if you look at models, and there's a specific model that was designed to look at this question by a group in Italy, Stefano Merler and colleagues, or you could use simpler branching process-like models, you get somewhere on the order of 5% to 60% for a flu-like infectious agent that's transmissible to a level comparable to flu. So if you multiply those probabilities, you get somewhere between 1 in 10,000 and 1 in 1,000 per BSL-3 laboratory year of working on, creating these infectious agents. Those should be adjusted. This is my point about this is not the final story. And they should be adjusted downwards by the things in green. Things like control measures, which are included in one of those probability estimates, but not in all. By the possibility that we could vaccinate or prophylax laboratory workers against infection, by the fact that these are in BSL-3+, which is indeed probably more secure than BSL-3, although we have a little data, and other strategies that may be employed in the future to control these agents. I will say that the repeated phenomenon with CDC mishaps of taking a infectious agent from a laboratory where it's used, or moving it in the false belief that it's been killed to another laboratory, is a very good way to circumvent many of these adjustments. And I think that's an important scenario to consider. On the other hand, everybody in the biosafety community believes that laboratory-acquired infections are underreported, and that I think it's fair to assume that the standards in the global laboratories are not all up to those in the US and the UK. So when you combine these, so then when you look at the consequence side, you need to multiply the expected attack rate in a pandemic times the case fatality rate times the global population. And for reasons of time, I think I won't go through that in great detail, but you end up with, for a highly virulent infectious agent, starting lower end being 1%, which is half of the 1918, and the upper end being 60%, which is what we think the H5N1 virus that infects humans from birds causes. You get somewhere between 20 million and 1.6 billion fatalities. This is a very much larger, of course, than any pandemic we have experienced, because the 1918 pandemic was in a much smaller world, and the more recent pandemics with this population size have been of much milder agents. These two should be adjusted, and perhaps we'll have time to discuss that. But if you take those basic estimates, and again, this is a first cut and not the final answer, you get a very large number. You get somewhere between 2,000 and, sorry, it should be 1.6 million fatalities. There's a rounding error there. 1.6 million fatalities per year of work in expectation. Of course, that's a low probability of a lot more than that, not a linear accumulation. But this is how we evaluate risk for low probability events, is we multiply probability times consequence. Okay, so that's the risk side. So what about the ethics of doing this? This is an underdeveloped field, and the ethical claims I am going to make in the next few slides are aspirational, in the sense that I don't believe that there is a consensus among those who think about bioethics or ethics in general right now that the ethical principles I would suggest applying to these experiments apply. And therefore, I don't believe that it was unethical to do them. I don't think that they violated any widely shared ethical principles. I think that the principles that they violate should become widely shared, but that's different from accusing. I don't want to be seen as accusing them of having done something that was unethical at the time, because I don't believe that you can outlaw things retrospectively, and I just don't think it's been thought about carefully enough. So my argument would be that there are principles that we agree on when we discuss the ethics of experiments on humans, on individual humans. And the bedrock of those principles is the Nuremberg Code, 2 points of which I've taken for this slide, and I think are compelling not just in relation to experiments that identified people that we can point to at risk, but in relation to experiments that put people at risk. There's nothing in the ethical underpinnings of the Nuremberg Code, in my view, that should apply only to human experiments, although it has been applied only to human experiments so far. So the first is that the experiment should be such as to yield fruitful results for the good of society, unprocurable by other methods of means or study. So I'm going to talk about alternatives in a few minutes, and that's where the unprocurable part comes in. And that the degree of risk to be taken should never exceed that determined by the humanitarian importance of the problem to be solved by the experiment. That is, there should be some predictable benefit that is not purely for knowledge, but is for humanitarian, which I would read as public health benefits. But all of these are interpretations that you may wish to disagree with. I want to bring to your attention something that I learned about because of this discussion about a year ago. This is a statement that all the major academies of sciences have signed up to, that I bet, I'm curious, has anyone seen this statement before? It's a very strong statement. It's essentially a Hippocratic sort of principle for scientists, that we have an obligation to do no harm, taking into consideration the reasonably foreseeable consequences of our activities. That's a very strong statement, I think even perhaps stronger than the Nuremberg Code. I'm not sure I even agree with it, but it is something that our academies of sciences have signed up to, so it's quite remarkable. So I will leave that there for the moment, and I hope we'll come back to it in the discussion. I want to talk briefly about the benefits, and I've abbreviated this part partly for time and partly because I think this will be the most fruitful, perhaps the most fruitful part to debate and discuss following Derek's talk, which I don't know what we'll say, but I think this will be an area where perhaps we can have a fruitful discussion. The major claim at the moment of those who claim benefits for public health is that there will be actionable predictions of a pandemic risk possible with the data that we obtain by learning in the laboratory how viruses adapt to become transmissible. So if we can identify the changes in the virus that lead to transmissibility, then we can find those changes in isolates from natural populations and make better predictions of which viruses we need to develop countermeasures for. And I'm going to argue that that's highly speculative, meaning not feasible in the medium term. The first reason it's highly speculative is that we don't see enough viruses to predict pandemic risk well. Over the last 6 years, there were about 1,600 outbreaks, 1,600 flocks of birds that were known to have outbreaks of highly pathogenic avian flu, almost all of which were H5N1. And in that period, each of the two databases that have reasonable number of flu strains in them got about one isolate from each of those outbreaks. And it's not actually one per outbreak, it's many from some outbreaks and none from other outbreaks. So on average, we're seeing one isolate, so one of the many genotypes in a bird, from one bird in one flock in each of these outbreaks. And because it's clumped sampling, it's actually not even one in each, it's one in each on average. And it takes over 6 months to a year before the sequences of those viruses become publicly available. So if every bird is harboring a dozen sequences in reasonable numbers, which is a decent guess, and there are many birds in each flock, probably harboring related sequences, but nonetheless, each flock of birds has many, many sequences in it, and we're seeing on average one, but in many cases 0. So we're not seeing everything that's out there. We're seeing perhaps the tip of the iceberg, the very, very most common strains that we happen to pick up. But that could be solved potentially with much, much more sequencing, although I would say it might have to be a whole lot more, and trying to figure that out would be an interesting problem for someone. A more fundamental problem is the problem of epistasis. In genetic terms, epistasis is the idea that the effect of a particular genetic trait, a particular base at a particular locus, in the case of a viral sequence, depends on the background in which it occurs. And there are multiple papers about the phenomenon of how widespread epistasis is in flu. And there are even more papers describing how a particular change that you might see in a natural isolate and either worry about or not worry about affects biological properties of flu. But what's remarkable is that on the left hand, the middle column shows what everyone thought was the phenotype or the effect on the biology. And the right hand column shows the exceptions to that rule. And there are maybe 2 or 3 examples in one column and 1 or 2 examples in the other column. So we're not talking about statistics here. We're talking about once in a while it's this and once in a while it's that. And we really don't know when we see a particular isolate what its risk level is going to be. or what its trait is going to be, because we've not studied it necessarily on that background. And as soon as Sean gave me the 5-minute mark, okay, good. And what we're really interested in is transmission, and even the simpler traits then interact to produce the more complex phenotype of transmissibility, which it in turn interacts with severity to create a level of concern for pandemics. So this is a very poorly understood system. This title is my candidate for obvious truism of the week, which is that unvalidated heuristics can be misleading. So I'm going to make the claim that not only are we potentially getting not much information from using gain-of-function studies to predict pandemic risk, we could be misleading ourselves. And the reason is because of epistasis. It's because what a sequence change tells you in one context may be wrong in another context. And that's, I would argue, already happening. The example I want to use is the E627K mutation in the polymerase B subunit, B2 subunit, which in several viruses listed here seems to increase both virulence and transmission of the strain. In 2009, we had a pandemic of H1N1, which you all remember. It killed somewhere on the order of 100 or 200,000 people worldwide. Not many compared to the numbers I've just been showing you, but a lot of people. It did not have this mutation. It had the E, not the K. So it had the low-risk version. We didn't know that strain was coming. There was no surveillance going on in Mexican pigs, and so we didn't pick up that strain, and we didn't have an opportunity to judge its risk level. But had we done so, and had we been looking at PB2 for this PB2 mutation, we would have said, on that axis at least, it's low risk. In the last 3 months, 2 public health experts have stood up and given examples of how they are continuing to use that as that mutation, which actually was known before gain-of-function experiments, but has been reinforced by gain-of-function experiments, to evaluate the pandemic risk of 2 other types of virus, neither of which we know what it does. So we don't know the phenotype in that background, but they are using this heuristic, which we don't know if it's good, bad, misleading, or not. I mean, we have 1 example in one direction, 2 examples or 3 examples in another direction. That's not statistics, that's anecdote. And so we may be up-regulating our concern about irrelevant viruses or down-regulating our concern about viruses that are important. I want to finish in a timely fashion, so I will just summarize my claims about alternatives, and we'll maybe get some time to discuss them later. There are a number of types of experiments we can do with flu, and an even larger number of things we can do to try to defeat the flu. Most public health is not basic science. Most public health is surveillance and countermeasure development. And there are lots of things that we can do that are technology-driven rather than knowledge of flu-driven. All of those approaches to trying to defeat the flu are safe. They don't create potential pandemic pathogens. The only 18 or a dozen out of the large number of flu studies do. And I would argue that there's an opportunity cost to doing those things instead of putting more resources and time and expertise into safe work. And I would then conclude on the point that if we have extremely dangerous work that can be done, and we have alternatives that are safe and that are at least as scientifically promising, although they address slightly different questions in most cases, that we should have a very strong presumption against doing the dangerous ones. So I'll stop there and turn it over to Derek.
We're very grateful to have a response from Professor Derek Smith, who is Professor of Infectious Disease here at Cambridge, and who's going to respond to some of the points that Mark Lipsitch made.
Yes, thanks for the invitation, and thanks so much, Mark, for coming, and also for your dedication in this area. I mean, we're all in this because it is such an important area. My conflicts of interest are that I was a co-author on one of the original papers that Mark talked about, one of the two papers that showed these airborne transmissible H5 viruses. I'm the director of a WHO collaborating center based here in Cambridge on emerging infectious diseases. And our sister laboratories are the two laboratories that did this work that caused the recent brouhaha, the Kawaoka laboratory in Madison and the Fouchier laboratory. Our work is primarily focused on seasonal flu with those laboratories, but of course we do other work with those laboratories as well, including some work pandemic, which is how come we are. Some people from my lab are on those papers. I should also say that Mark and I have been friends for many years, and I've admired his work very much over that time. And I also think, and we recently co-authored a textbook chapter together, voluntarily, which we chose each other to do that chapter in. And although we very much agree on the need to do risk-benefit analysis, we are, at some way, extremes in terms of how the risks are and how the benefits are, exactly as Mark said, that there are people who, of course, we all understand that there are risks and benefits to be traded off. And as much as we can do this quantitatively, we would all like to do it more quantitatively. I also think it's really great that what's happening here is this is being done in a scientific way, in the sense of, papers are being published on this. We can have debates on this about facts. These are not decisions that are being made by fiat behind closed doors as to whether these experiments should be done or they shouldn't be done. And of course, it's incredibly important in science that we do things openly to keep scientists engaged on all sides, and the work that you do and the way that you publish it very much encourages that. In some sense, we might say that your summary is knowledge can't help. And I know you wouldn't say it that extreme, but it is a pessimism about how knowledge that can be gained, that can be learned from gain-of-function experiments, that it can't help in a domain that we know is very important, and that it's too risky to find out whether or not we can learn enough to actually have an impact to mitigate these diseases. And of course, this is a perfectly valid position. And of course, the context, the reason why Mark is dedicating time into it, and many other laboratories do as well, is because of the incredible importance of this question. About 15 years ago at the World Health Organization, flu experts from around the world came together and decided that figuring out whether or not avian influenza viruses could start transmitting between mammals, to try to assess what the risk of that was, is one of the most important questions for influenza public health. And all of that funding that Mark talked about in all multiple countries, funding the best laboratories in those countries and in other countries worldwide, is to answer that question, because it is such an incredibly important question. And during that 10-year period, we sort of thought that these viruses could actually start, that avian flu viruses could transmit between humans. These highly pathogenic H5 viruses that are subject of the experiments that Mark particularly talked about. And yes, the best scientists tried to figure out if this was true, and over that period, all attempts to do this failed. How can you know whether or not these viruses could actually transmit between humans? Well, the only way that anybody knows how to do that is to see what it would take to make them transmit between humans. And that's what all of these gain-of-function experiments try to do, is to take these avian viruses and see if they could be made to transmit between humans. Because if they can't, we should stop worrying about them. Now, the amount of money that we can spend to prepare for influenza pandemics, you heard from Mark how devastating they can be. We should really, our government should be paying an awful lot of attention to preparing us for one of those pandemics happening in nature, to either try to prevent it before it happens or to mitigate it if it does, if there is really a risk that they can happen. If there is not a risk that they can happen, there's a lot better things to spend those enormous amounts of money on. Not enormous amounts of money to find out, but enormous amounts of money to be prepared, to stockpile the vaccines, to stockpile the antivirals, to go through all of the international procedures in case a pandemic actually starts, which had happened for about a decade period from around about 1998. And there came a time during that period where people, sensible people, seriously started to say this is unlikely to happen. We've had 10 years of these viruses circulating in birds. They haven't started transmitting between humans yet. It's not going to happen. We should stop being so worried. We should stop spending all this money. You know, good papers were written in good places to say this. And then there was a breakthrough. There was a breakthrough of the two papers from the KNAW laboratory and the Fouchier laboratory that actually created viruses that could avian viruses that could transmit between mammals. Things that people had kind of figured couldn't be done. In fact, it only took about 5 mutations, and a couple of those mutations were already in nature. A third of those mutations is occasionally in nature, so 2 or 3 mutations. In our laboratory here, we figured out what's the chance of 2 or 3 mutations occurring during the course of an infection in a human so the virus could go on and transmit. And we knew that we didn't know enough and know it accurately yet, but from what we could estimate, we could say that there is a reasonable chance that that could happen, although the chance is low. And most importantly, we figured out from that work what experiments would be needed to be done to really refine that risk estimate, the risk estimate, not of the virus getting out of the lab, as Mark presented, but of the virus coming out of birds and getting into humans, the risk of nature doing what Mark talked about here. We know that nature has done that many times before over history. For flu, it's done it 4 times in the last 100 years. So that's sort of the background in which those experiments were done, and the breakthrough that Mark talked about in figuring out what it would actually take. Gain-of-function experiments to figure out what mutations it would actually take. I'm not going to go into the calculations of the risk. Mark is absolutely right. The risk, you know, exactly what that risk is, we don't have to know it precisely, but we do have to know it approximately where it is. And there are 2 probably extremes on that. There's the one that Mark published and talked about today. And then at the other extreme, there's one that comes from Ron Fouchier that is in press, that, as you said, is something like 6 orders of magnitude lower. And the primary reason why that the main difference there is to take exactly the same method and procedure that Mark produced, but to talk about the laboratories and the context in which the particular gain-of-function experiments were done in, and the acknowledgement of the fact that, yes, there are human errors. Systems have to be designed in order to take account that there will be malfunctions in procedures and that there will be malfunctions in machines, and so there are multiple layers of protection. For example, as Mark said, one can vaccinate the people who are in the lab doing these experiments. They have very high titers, which means that they're very well protected. Even if they do acquire an infection, they're unlikely to get a very much of an infection, so they're unlikely to transmit it. There's also procedures in place that if there is anything in the laboratory where somebody might have been exposed, and this is all highly reportable. If anything happens where where something happens in the laboratory, even if there's really such a tiny risk of exposure, it's reported. That person is put on prophylactic antiviral drugs that are highly active against flu, and also they're quarantined. These are multiple layers precisely because Any individual layer is flawed. Now, as Mark said, there is now a US government contract to try to also assess what this risk is. And it's a very important thing to figure out what the risk is. And we see where that risk actually comes out, and it's a very important thing to do. It is interesting to note, and when we think about what the risk is the fact that experiments on these types of viruses has been done for decades, and there hasn't been a release. It is interesting to think about the one in 1977 that Mark talked about, where for many years people would say anecdotally and colloquially that the 1977 flu pandemic was caused from a laboratory accident. Well, in 1977, this is a time when there were very little security in the laboratory. Things were basically done on the open bench. It's also a time, and from a part of the world where that happened, where people were also doing experimental infections in humans of attenuated vaccine viruses and wild-type viruses. The idea of a laboratory release causing that is a very different type of laboratory and a very different situation than through today. But all of this will be refined, and fortunately it's going to be done in a way that it can be peer-reviewed by people and followed up by people who think one way or another in all of these areas. The other thing to think about, and the key thing, is what the benefits might be. And there is actually an example, I think, that we can go through on this. And that is the example of the H7 influenza viruses that have been circulating in China in birds and have killed tens of humans over the last year or so. And this, I think, is really a very good example of the good and the bad of what has been happening. China was very quickly, we think, very open about those H7 infections. And from the very early on, they were reporting about those H7 viruses that were isolated from humans, that they had specific changes in the receptor binding site that had been identified by these types of experiments in the past. The PB2 substitution that Mark talked about in terms of the dangers of heuristics, and that they also didn't have a particular they lost a glycosylation site close to the receptor binding site, which is another one of these things that came out of these gain-of-function experiments. And perhaps, although we don't know, the openness of reporting about these viruses is because of the warning signs that they had. They could look for these genetic markers. But of course, Mark is absolutely right that those genetic markers were determined in other very specific viruses, and we don't know yet how much epistasis there is, what is controlling that epistasis, and whether or not those particular markers are relevant for why those H7 viruses in China, those avian viruses, have been infecting humans and killing tens of humans. But they do give us an idea that it is something that we should look at and we should pay attention to. And that happened. In the US, for example, within a couple of days of these viruses being reported, vaccine candidates were being synthetically generated, vaccines being made and started to be made in order to protect in case that pandemic happened. And that doesn't say that we know that those viruses are easily transmittable between humans, but what it says is they may be. And that's one of the places where we should spend tens of millions of pounds to be prepared. Now, of course, there may be other ones that we don't know about yet. And now what's critical is what one does next. And this is the other part of the story that I want to tell, is that within a couple of months, we could have known whether or not those substitutions were important. Within a couple of months, we could have known, if it wasn't those substitutions, which ones would make those viruses more transmissible between mammals by doing exactly the sort of gain-of-function experiments that Mark talked about that have been done for H5. We could have known that once they'd fully adapted and could transmit to humans, among humans, using the ferret model, whether or not they had changed antigenically and if the vaccines would still work, And if not, how they would likely change antigenically, so what different vaccine we should make. We would know whether or not they would be susceptible to the different flu antivirals that we have. And we could know how pathogenic they are in ferrets, which is a decent model for humans, although not perfect. We could have known all that trivially within a couple of months. We know nothing of it. We know zero. Those experiments have not been done. We are in a situation where we know that there's a real reason for concern of those viruses, and tens of millions have been spent. We know that there are those markers, but we don't know whether or not they are for real. But we could absolutely know for those viruses. And if we did know that those markers were for real, and we saw another, and we knew which other substitutions, which may be the same as we've seen before or may be different, we saw another one of those come up, and we say, oh my goodness, there's really a risk. There could be massive culling of poultry in China to stop this virus circulating in birds, and thus the risk of getting into humans. We might have stopped a pandemic. That is the benefit, as I see it, and as many people at the World Health Organization and people who work in this domain see it, of what really could have been done. And the reason why zero of it could have been done is because of the fact that after these experiments, there was a moratorium worldwide on gain-of-function experiments. And then new procedures put in place for regulating exactly what experiments could be done next. The sort of things that Mark talked about that are there for smallpox. These regulations are already in place, but were already in place beforehand, but going through them, a much more drawn-out process now. And before the current pause, which came in 2 months ago? In October, 3 months ago. Before the current pause, it had been a year where a couple of laboratories worldwide had applied for permission into those new regulations to do these, the experiments that would have taken a couple of months to know all this stuff, and that permission had not come through yet. Those experiments are no more dangerous than ones that have already happened. They're not done in different laboratories from ones that had already happened. What's key, and now with the pause that will probably go on for a year, they won't be done for another year. This is the other side of the coin. This is the other risk of not doing the experiments. We basically have, although we don't have to be ostriches and have our head buried in the sand about those viruses, we do. We don't know whether or not we should be spending hundreds of millions of dollars or billions of dollars to prepare for a pandemic of those viruses. We don't know if we should be culling all those chickens in China, which nobody wants to do, because they're a major source of protein for many people and also in other parts of Asia and a critical part of the diet. Yeah, well, we don't know that. And there are other viruses that have popped up. As Mark also said, flu viruses change all the time. No, as you said, flu viruses change all the time. The other ones that have popped up, like the H5N6, we know very little about those viruses. We could and we're not. So one of the things is, in this period where there is doubt about whether or not knowledge can help in this area, should we keep going or should we stop? Is it really too risky to keep going? Keep going in the way that we've been going for decades without these viruses getting out of the laboratory? Should we really stop while we try to do a better risk assessment? Nobody would say that a risk assessment like is being done now shouldn't be done. But the cost of this is potentially large. The cost is potentially zero if these viruses don't start circulating among humans. But we know that these viruses, that flu has done this in the past and has circulated among humans and can be devastating. The numbers that Mark gave of how many people might die from a laboratory release of a virus, nature can do that too and has. So we have to balance, when we do this cost assessment and this risk-benefit analysis, we have to balance all of the risks and the risks of not doing the work as well. And it is also kind of interesting that, that in some sense, we could use, so I think we're in a situation where we should say, should we continue now, and should we pause, or should we, and evaluate, or should we keep going and evaluate? Well, we know that nature is not going to stop. And if we stop, as we are, then we are not going to see whether or not know how well we can mitigate these pandemics. There is one other thing as well, and that is we have 2 laboratories that are waiting for permission to do those experiments on H7. And I think also, as well as I honored and respect Mark for what he's doing, which I know he's doing for existential risk reasons, I think it's also the work that happens in Ron's lab and Yoshi's lab. I've known them for many years, and they are also, as are many others, trying to do their work also for existential risk reasons. And I also want to honor them for a moment, because they are the 2 labs that have, or 2 of the labs, and I think perhaps the only 2 that are waiting for permission to do the experiments on H7. And they do this among death threats from all of the media attention that does with this, and not just to them, but to their families. And for me, I think there is also a great deal of to see them continue to do the work that they think is important, and that all colleagues at the World Health Organization think is important as well. In the face of that, I think is also a very honorable thing to do. So that's it, and I think we leave time for mostly to have debate, discussion.